Hi everyone, my name is Nethra Srinivasan, and my research project is the effect of the onset of menopause on the transcriptomic profile of the aging female brain. There appears to be a divergence in the occurrence of neurodegenerative diseases between males and females, but the mechanisms so far are unknown. For example, for Alzheimer's disease, more than 5.5 million Americans are living with Alzheimer's disease, of whom 2/3 are elderly females. In addition, the mitochondria of younger females are protected against the pathologies of Alzheimer's disease, such as beta amyloid toxicity, reactive oxygen species, and apoptogenic signals, compared to their male counterparts. However, this advantage is lost in the mitochondria of elderly females. And the difference between the younger females and the elderly females is this transition into menopause. And in addition, current Alzheimer's treatments only target acetylcholine and glutamate neurotransmitters in order to treat the symptoms, but they are not treating the root cause. And so, because of this discrepancy between the two genders and this transitioning into menopause, it's important to figure out the underlying causes to be able to better target treatments. So, I will be figuring out what is going on as a female ages and if there are any transcriptomic differences to identify the genes at play, as well as pinpoint pathways to better target treatments. First of all, to give some background information about female aging, menopause is the end of a female's menstrual cycle and fertility, in which the ovaries no longer produce estrogen and progesterone. In this project, I will be looking at data obtained from rats and rat menopause. So the ages that I will be talking about here are in rat age. So the first stage is premenopause, which is at about 6 months for rats, and this is when they have normal menstrual cycles. Then they transition into perimenopause at around 9 months, in which the cycles are irregular but they have not stopped completely. Then they transition into menopause at about 9 months, which starts with the final menstrual cycle. And then at around 15 months, they will be in postmenopause, which in humans, this is the year mark from the final cycle. Now, to continue on about a little bit of background about the aging of the female brain, as the brain ages, it's shown that there is an increased immune response. And this is obtained from Mishra et al., from which I am using the data from. So for the dataset distribution, I'm using transcriptomic data from the rat female hippocampus, and there are 6 groups with 6 samples each. So there is the female rat at 6 months, or reg 6M, and then at 9 months, normal menstrual cycle, or reg 9M, then 9 months irregular menstrual cycle, which is irreg 9M, 9 months acyclic menstrual cycle, acyclic 9M. Then OVX at 9 months, which is overrectomized. And overrectomized basically models the most important clinical features of estrogen deficiency induced or postmenopausal conditions. And this way it acts as a control for my experiment. And then there's also the female rat at 16 months, acyclic menstrual cycle, or acyclic 16M. So my research question and hypothesis, I'd like to understand the genes expressed differently in the progressive stages of female aging and menopause, which can help us understand the risks for neurodegeneration and pathways to target in order to treat neurodegeneration, such as for Alzheimer's disease in women. So the different ideas that I'm looking at are how is the age and time of the onset of menopause affecting the transcriptome expression and which pathways are affected? And are there biomarkers in menopausal women that can help us identify their risk for certain neurodegenerative disorders? Or how does the expression differ in overrectomized specimens and regular specimens? And can this lead to insights on the onset of neurodegenerative disease? And my central question is, what are the effects of the onset of menopause on the transcriptomic profile of the aging female brain? So I hypothesize that as the females enter premenopause and perimenopause, they'll express less genes related to neurodegeneration as the females in menopause and postmenopause. And basically, that the age and the age of the onset do have an effect on the transcriptomics of the rat. So for my methodology, since there's 6 groups to compare, I'll be using principal component analysis, DSeq2, which is differential gene expression analysis, and hierarchical clustering in order to relate the expression to each other and draw conclusions, analyze which genes are overexpressed and underexpressed in the 6 groups, and look at the significantly expressed genes to annotate their function, and David and Richard to see the pathways that are expressed. So firstly here, with my preliminary PCA, I found that there appeared to be a divergence between the regular groups and the irregular and acyclic groups. And therefore, I grouped it into regular versus acyclic and regular versus irregular. And as we can see in the PCA, there are 2 different clusters for regular versus irregular, which shows that they're different. And for the regular versus irregular, there does not appear to be 2 separate clusters, but that's only because the components are very small. But once I did differential expression and I did the PCA again for the separate significant genes, I found that there appeared to be 2 distinct clusters. And also to test the control, I did PCA between the regular versus OVX, and I found that there were 2 different clusters, showing that there appears to be a divergence when there's the shift into menopause, and it cancels out for the age. And continuing on for my differential expression, I obtained some plots such as these volcano plots, which shows the significant genes. And I also found that the pathways that were upregulated in both the regular versus irregular and regular versus acyclic were functions related to the immune system. And the pathways that were downregulated were related to mitochondrial dysfunctioning, such as oxidative phosphorylation and the mitochondrial respiratory chain complex, which in turn leads to the downregulation of the pathways for neurodegeneration, such as Alzheimer's, Parkinson's, and Huntington's. And for further hierarchical clustering, I found that there appeared to be 2 different clusters, which shows that there is a discrepancy between the regular versus acyclic and the regular versus irregular. And I did pathway analysis with the functional annotation, and I have images over here showing the relative expression of the upregulated and downregulated pathways. So here's upregulated immune system, downregulated related to the mitochondrial function and neurodegenerative diseases. Same thing over here for regular versus acyclic. And so for my conclusion and discussion, I found that there is an impact on the transcriptomics of the rat, especially in relation to the mitochondrial function leading to neurodegenerative diseases such as Alzheimer's and Parkinson's and Huntington's. I also found that the immune system is heightened through this data. And this helps me say that the pathways that should be targeted for possible treatments of neurodegenerative diseases should be in relation to oxidative stress. And this also supports the current pathologies for Alzheimer's, in which the accumulation of the beta amyloid plaques exacerbates the oxidative stress, which damages the mitochondrial structure, which shows that through targeting menopause, we can possibly be able to find certain treatments, because menopause appears to be a factor in the onset of neurodegenerative diseases. Now, for my limitations, I was not able to extract this data myself in a lab, and I had to use the data that other researchers extracted from their samples. So I was not able to control the sample size or the procedures used to collect the data, and therefore I'm dependent on the collection strategy to obtain my results. And for the implications, by now that we through my project, we can see that menopause is a factor on the oxidative stress, which leads to these neurodegenerative diseases. By being able to remove the cause of the oxidative stress, according to Cole et al., the oxidative stress, it creates damage on the mitochondria, but once the cause of the oxidative stress is removed, the mitochondrial membrane is actually able to rebound and maintain its structure again, which could possibly help us with targeting for neurodegenerative diseases and could be revolutionary to the field. So thank you for listening to my presentation.